Someone know how this thing is working? Okay, it was laptop. Laptop is connected. On is on, it's okay. מה נשאר? לא, זה המחשב של זה.
מה קורה?
עכשיו.
עכשיו? שלך?
עכשיו נראה.
אתה יכול הולך להציג את זה?
What time is that? Sunday morning at 9 o'clock in the morning? You should protest. I mean, for me, what you want, okay, but you? 9 o'clock in the morning, Sunday? I have to talk with Mati about this. Mati asked me to talk about the cerebellum. And how many of you have been to the conference last week? What? Yeah, Michael?
Do we go?
Better? Okay, so how many of you have been in the meeting conference, What Makes Us Human, last week? And I saw that there was many people talking about the crown of evolution is the cortex. Only few said something, oh, maybe we should talk about cerebellum. But I want to start the talk of a few slides of what is a cerebellum and why it's important. And my first slides refer to a work published about 6-7 years ago, which called Life Without the Cerebellum. And this work was done by Boyd, that actually is a tribute to his grandfather, I think, or maybe his father, that did a work in the 40s in London. And it was a patient, HC. And HC lived to the age of 76. At 40 to live to the age of 76, this is by itself an achievement. And after he died, where absence of cerebellum was discovered. Not cerebellum at all, and he lived to the age of 76, at 40. And that led to the myth that people who are born without a cerebellum may have no observable symptom at all. So actually, it's useless. That's the meaning, the direct translation. Have no observable symptom, meaning it's useless. Now, from the evolutionary point of view, as we heard last week, to have billions of neurons that are useless, that's very unlikely. But this is not the end of the story. That's, we have another paper published about 4 years ago, where now the people are still alive, And you can see the MRI, and this is the place of the cerebellum. There's nothing, absolutely nothing. But unlike the 40s, we know exactly what the symptom. It's a she. She had never learned to read or write. Her speech developed later on her slide. She showed poor dexterity and severely disrupted predictive control of object class. She also showed evidence of acquiring conditioning eye blink, this is not important, and showed mild to moderate, mild to moderate neurophysiological impairment in IQ, planning, behavior, visual, verbal, and spatial memory, perception and attention. So basically, she didn't have children, but she got married and have children. But she have all these symptoms that are a sign of a missing cerebellum. So, but you can live without the cerebellum. It seems to be that you live very. To make the story out somehow balanced, what about living without whatever? What about living without cortex? Now, we don't know of any cases where living without cortex, but our people in genetics manage to make a mouse that doesn't have any cerebral cortex. Nothing at all. Honey provide normal singing. And if you look at the slide, there's a sagittal section through the brain of the mouse. This is a normal mouse with a cortex, hippocampus. This is the mutant one. All these things is missing. And it sings. It means it's very happy, no? So we don't need the cortex. But, I look at this kind of studies and other studies like missing cerebellum and birth, and what actually we learn from this kind of experiment. We don't learn what the structure is doing. We do learn how the system manage without this particular part. So all the stories of knockout things, you knock out a molecule and you say, oh, this molecule is important, it's not important. This is nonsense. What we study under this condition is how the system, which everyone will agree, the basic, most important feature of the system is plasticity. How this system can handle huge range of problems without observable symptoms. And the last thing is evolution. And we heard, I said at the meeting yesterday, that what makes us human, it's the development of the cortex and cerebral cortex, and there is everything is taking part. There were a few people in the meeting that said maybe the organizer should invite someone who do talking about evolution of the cerebellum. And I brought you this paper. This paper, A Rapid Evolution of the Cerebellum in Human, in other apes. And in this study, what they do is human unique cognitive ability. I'm reading what's in the paper, what's in there. Human ability are usually attributed to a greatly expanded neocortex, which has been described as the crown achievement of evolution and the biological substrate of human mental ability. The human cerebellum, however, contains 4 times more neurons than the neocortex and is attracting increasing attention for its wide range of cognitive function. So this is the introduction for the problem. Then they go and they do method to describe evolutionary rate change along branches of phylogenetic trees. You know this thing that you can show how species evolve from all kind of ancient parents. We showed that the cerebellum underwent rapid size increase throughout evolution of apes, including human, expanding significantly faster than predicted by the change in neocortex size. So when you look on evolution and you look only the cortex, you miss the cerebellum. The cerebellum evolved much more if you compare the development of the cortex to the cerebellum, more and faster than what we have in the cortex. And I conclude that cerebellar specialization was a far more important component in human evolution than previously described. And again, I go to my basic idea. You cannot generate billion of cells to be useless. I'm coming from a biological background. In biology, I claim that evolution, one of the things that drive evolution is the need to save. So to generate billion of cells to do nothing, this is very inefficient way for evolve. So just so the cerebellum is very important, but just to make the thing a little bit balanced, I brought you that the last paper saying the elephant brain. And we look at the elephant brain, and the elephant brain have 257 billion neurons, 3 times more than the average human brain. Over 97% of the neurons in the elephant brain are found in the cerebellum. 97% in the cerebellum. And we cannot say that from cognitive point of view the elephant is more evolved than the human brain. But if you look at the picture, you see that this is the human brain, and this is the African elephant brain, and this is the cortex of the brain, and cortex of the human, and the cerebellum of the human, and this huge thing is the cerebellum of the elephant. And yet, the cortex of the elephant have less cells than the human cortex. It means that maybe there is something in the number of cells in the cortex of the human that is smaller than the cortex of the elephant, but yet have more cell in there. Maybe this is what contributes to the hypothesis that the larger absolute number of neurons in the human cerebellar cortex, but not in the whole brain, is correlated with superior cognitive abilities of humans. So, what's the bottom line that I want you to take home? It's counting the number. It's useless. It's meaningless. It's not important. Connectivity might be more important, but all these people, what they can do, they can count the number of cells. That's what they know. They got to 200, 300 billion cells. Meaning what? Nothing. We'll go back to the issue of evolution a little bit later, but right now I want to give you some background on the organization of the cerebellar cortex. So, schematically, we make a cut in the cerebellar cortex, and this is the horizontal axis, and this is the coronal axis of the cortex. And we see the major component there is the Purkinje cell. And the Purkinje cell is amazing cell. From all the cells that I know, first is one of the largest cells in the brain. From all the cells that I know, it's the only cell that's organized in 2 dimensions. It's flat, completely flat. So if you look at it from this view, you see 300 or half a millimeter long and wide. If you look at it from the other side, it's 5 micron, 5 micron width. It's like a fan. It's like, you know, those who have been in coral reef diving. Corals look like that. And they organize just perpendicular to the stream of water. And they do that in order to capture most of the food. Because if they are perpendicular to the stream, they catch most of the food that they can. And here, this is organized in this way to get the most of all the fiber that's going through this area, which called the parallel fiber. We'll get to that in a minute. We have one layer of Purkinje cell. And Purkinje cell is, the number of Purkinje cell is only 5 or 2% of the cells in the cerebellum. But they are unique in their appearance, and I hope to show you some other figure. They organize in a single layer, the cell next to another. There's nothing in between. They organize single layer, and the output is the only output of the cerebellar cortex. So if something happened in the cerebellar cortex, and probably something happened in the cerebellar cortex, the rest of the brain know what happened in the cerebellar cortex through this output. The output, the axon of the Purkinje cell. The major problem in this is that the Purkinje cells are inhibitory neurons. They release GABA, and they inhibit the next station. That's the only thing they can do. And the major question is how you translate inhibition into output, because you want the rest of the brain to know something of what happened in the cerebellum. So you have to translate this inhibitory input into some excitatory input, so the rest of the brain will know about that. So these are the Purkinje cell. The other, the 2 input to the Purkinje cell, one is the climbing fiber. And the climbing fiber are the blue color lines here. And it's also a very unique structure compared to the rest of the brain. Each Purkinje cell receives a single climbing fiber. One presynaptic axon. One fiber delivers the information to a single. There's no other climbing fiber to this Purkinje cell. There are several more Purkinje cells that receive the climbing fiber. So a single climbing fiber may innervate several Purkinje cells, but each Purkinje cell has a single climbing fiber. Not only a single climbing fiber, as we'll see it, this is one of the most reliable, efficient synaptic contact in the brain. Once the climbing fiber is firing, Purkinje cell is forced to follow. There's no other way. It cannot avoid it. There's no probability. There's no need for integration. There's no need of summation. It fires, the Purkinje cell fire. This is one of the strongest synapses described in the brain. The other input is the motif fiber. The motif the fiber coming from all over the brain, especially from the brainstem area. They a group of huge number of axons coming into the area and innervate a group of neurons in the cerebellar cortex that's called the granular cell. So the granular cell occupies the a very large area, and these are the reason for the huge number of neurons that we have in the cerebellum, because they very small, they fill up the entire what's called the granular cell layer. They fill up the entire area there. They are going up to the other side of the cerebellum, to the outer layer, and bifurcate. And thousands of billions of neurons, granular cells, generate this huge area of neuron that actually innervate the Purkinje cell. It's an excitatory pathway that innervates the Purkinje cell. So each Purkinje cell receives one climbing fiber and about 100 to 200,000 parallel fibers. So a neuron at one side sums a lot of information, and on the other side receive a single input. Of course, each synapse of the parallel fiber is very weak, and in order to respond, it has to sum up a lot of information, a lot of fiber, in order to trigger a response in the Purkinje cell. So we have a beautiful situation. One climbing fiber innervates one cell, and another 100,000 parallel fiber innervating the same cell. And I think this is the reason for the very old idea that the climbing fiber serves as a teacher, and all the parallel fiber are students. And the teacher is coming and making the whip and make sound. Everything is becoming very quiet. It's true, it's become very quiet. But then, during this process, all the students start to learn something. And so the idea is the climbing fiber instruct the Purkinje cell how to respond to the parallel fiber. And this is the teaching system, and a lot of work has been done demonstrating the interaction between the climbing fiber input and the parallel fiber input. And if you pair together the parallel fiber and the climbing fiber, you induce long-term depression in the parallel fiber Purkinje cell input. So this is a kind of a teaching system, and the idea is that the climbing fiber is a teacher in a simple way. It's an error signal. Says something gets wrong, let's change it. That's the point of this organization. Of course, like in any other system in the brain, we have some inhibitor interneuron in the cerebellar cortex. The largest one is the Golgi cell, and you see the Golgi cell receives the input also from the parallel fiber and deliver the output to the granular cell. This is one of the largest axon bifurcation that is known. I always wonder why people spend so many times to identify the dendritic morphology and how many bifurcations and how many spines. Nobody is taking care of this axon. Look at the axon of the Golgi cell. Huge. I know some cell in the cortex also. Huge number of axonal terminal. Axonal terminal that terminate on this granular cell, inhibit the granular cell. So you have a feedback inhibition like circuit there that, but look at the axon. It's huge. It's a huge area. And nobody study in detail the axonal ramification as they measure the dendritic one. And it's another problem, and I don't see why. Of course, in addition to the Golgi cell, we have another two type of inhibitor in the interneuron there in the cortex, One is the basket cell, the red one, and it's called basket cell because you see the axon going and wrapped around Purkinje cell soma, like a basket. Extremely powerful inhibition. Once this is activated, Purkinje cell cannot fire because it's strongly inhibited. Because the synapse is very strong, because it's located in strategic place, so it prevents Purkinje cell from firing. And the other one is the stellite. And the stellites innervate mostly the Purkinje cell dendrite. So the two inhibitory systems within the cerebellar cortex, one to prevent the cell from firing, and one is to participate in integration along the within the Purkinje cell dendrite. Two inhibitory systems. This is the cerebellum of mammalian, and the amazing thing about that it's extremely preserved through evolution. So if you look on frog cerebellum, and you see frog cerebellum is a tiny little thing there, and within this tiny little thing you have Purkinje cell, a flat two-dimensional Purkinje cell. You have parallel fiber, you have climbing fiber, you have the entire circuitry there. And you move higher in evolution. And you move higher in evolution to the alligator. So from frog to reptile. And we see exactly the same thing. The cerebellum is a little bit bigger. The Purkinje cells are there. Parallel fiber are there, climbing fiber, everything is preserved exactly the same. And you can move on to the pigeon, and the pigeon have a huge cerebellum. The idea at the time was that cerebellum, it still is, the cerebellum is involved in movement and organizing the movement. And the birds has to organize the movement in 3D. We have to organize the movement in 2D. That requires a much larger cerebellum. That's the idea of the big cerebellum of the birds. And finally, the cat cerebellum, which is also very much similar, following the same concept. Climbing fiber, parallel fiber, Purkinje cell, and a bunch of interneuron. That's all we have in the cerebellar cortex. And that's evolved in exponential rate through evolution. And we end up that in human, we have at least the same number of neurons in the cerebellum than in the rest of the brain. In elephant, we have more than neuron in the cerebellum than in the rest of the brain. But it's continued to evolve, and my interpretation is that it's a system evolved, and they're doing extremely well job, a very good job of doesn't matter what. And then in evolution, it's very difficult to invent everything from start. So what they do, they take this thing and they expand the implication. So if you use it in the alligator or in a frog, you use it to coordinate movement. In a more advanced animal, you can coordinate other things. And you need coordination. You need to coordinate your attention, for example. You have to pay attention to something at a given time. If you don't pay attention to one thing and you constantly bombard by sensory information, you have to get crazy. You cannot stop things and be focused on one. So coordinate the activity within the cortex, within the forebrain, is a challenging task. And we have a system evolved over the year to make coordination. We can use it to do some other thing to coordinate attention, state, mind state, cognitive function, whatever you want. And you will see that some people think that the cerebellum really does all this. Now, that's what the cerebellar cortex, but the cerebellar cortex covers what's called the cerebellar nuclei. And the third member in this party is the neuron of the inferior olive. Neuron of the inferior olive are the source of the climbing fiber. So people define something that's called cerebellar module. The basic unit of the cerebellum is not unit within the cortex, is unit within the entire structure of the cerebellum. And the module is something that appears itself. It can be shown to occur all over the cerebellum. And what you see is that the Purkinje cell is one component in this module. The cerebellar nuclei, and the cerebellar nuclei are the only output of the cerebellum. So if anybody in the brain knows what happened in the cerebellum, is through the cerebellar nuclei. And we have here, you can see we have two types of neuron, but it's only two types presented here. We have many types of neuron within the cerebellar nuclei, but I'm interested in these two right now. And then we have the inferior olive. As I said, the inferior olive is the source of the climbing fiber, this powerful input to the cerebellar cortex. And the module is made, the wiring diagram is that the inferior olive goes all the way to the cerebellar cortex, generate the climbing fiber synapse, as I said before. The output of the Purkinje cell goes down to the deep nuclei. Now, in the deep nuclei, the small neuron, this one, is also inhibitory neuron that go back to the inferior olive. And this generates a closed loop of one excitatory part and two inhibitory parts. Inhibitory of the Purkinje cell, inhibitory of the cell, and we will talk about that in hopefully today. And the last part is, of course, this big blue deep cerebellar nuclei cell that transmit the output of the cerebellum. Again, if somebody knows if anything happened in all this huge and really huge system, if anybody pay attention to what happened there, is through this path.
The connection between the Purkinje cells and the cerebellar nuclei is excitatory?
Inhibitory.
So you said that the output of the cerebellum is from the cerebellar nuclei only?
This is excitatory. The Purkinje cell is inhibitory. And this one is excitatory. So, you take a break, right? You want a break. You need a break. Before we go into the break, I will just start with a simple question. What's the function of the cerebellum? Billion of cells. You have to do something. So what you do these days, you want to know what the cerebellum is doing. So you go to the internet and you press cerebellar function. And you got billion of papers about cerebellar function, and I did some job for you, and I collect this one, something about movement, human locomotion. It's many papers about cerebellum involvement with motor task. Then we have another group that's talking about sensory role of the cerebellum in sensory perception. And then we have another thing that can say, well, it's a completely different. Autism in the cerebellum. Autism in the cerebellum is today a very hot item because, people think that if autism, it's the people that suffer from problem of coordinate their attention. Cerebellum is perfect for that, but it's not only wishful thinking, and there are very strong evidence that cerebellum is deeply involved in some of the autism disorder. So what do you do? Now, if you are not in the field of the cerebellum, you have to laugh. I mean, people study this structure over the last 200 years, they still don't know what it's doing. If you are inside the field, my God, we studied this thing for the last 200 years, we still don't know what they do. So you can either laugh or cry. The problem is that you are still stuck in the cerebellum. And this is a real thing, pathological laughter and crying. And it's a link to the cerebellum. He said, this is funny. Okay, good humor. Okay, good humor. You go back at that. Humor comprehension. Link to the left cerebellum. So you cannot get out of it. Any point you go, it's the cerebellum. The cerebellum is there, and we don't know what it does. I think it's exaggerating to say we don't know what it does. We know something about what it does, and we will start with this example that I like the most. And this is a work done on the cerebellar patient, and I give him a very simple task. They have to throw a ball on a target, and the dots are the points where the place where the ball hit the target. So you're sitting there, you have a ball, you throw the ball on the target, and you mark the target. And these are normal people, and these are cerebellar patients. Now, I'm not sure I can do that good, so it's not nice to compare a professional ball thrower to a cerebellar patient, but the idea is there. And the point that the people observe is that the ball hit the target, the spread of the points are along the y-axis, much less than the x-axis. So you either hit too high or too low, but not that much sideways. So what's the cause of this bad performance? Now it's people, what they did, they measured the trajectory of the hand movement while we throw the ball. So you hit the ball, and we are throwing the ball, and we looked at the trajectory of the hand. You look at the trajectory of the hand, and you see these are the professional balls, and these are the cerebellar thing. Now, you can see that this is a little bit more diverged than this, but not by much. This cannot explain this difference. So what does explain the difference? What does explain the differences is the timing. The timing of when you throw a ball, you take the ball, and at a certain point along the path, you release your finger and let the ball go. The timing of open the finger, let the ball go, marked with the dots here. And this is the huge difference compared to what we have in normal people. So what's missing in this cerebellar patient is a timing signal to tell you when to open the finger and let the ball go. And it's obvious that when you let the ball goes early, you will hit high. And if you let the ball go late, you will hit very low. And this will explain this spread of the results. So according to this paper, is what's missing in the cerebellar patient is the exact time of open the finger and let the ball. Timing is important. But when somebody tells me time is important, of course timing is important. That's what the brain knows how to do. It can only play with time. There's nothing else. It doesn't play ball. It plays with time. It generates signal at the right time. It composes a pattern of signal at the right sequence. That's what the brain knows how to do. And with this ability, you have to do all this fancy function. So I'll finish with the last demonstration. And the question arises several years ago, why can't we tickle ourselves? You know that we cannot tickle. It doesn't feel the same. We just cannot tickle ourselves. And these people I was interested in studying, why can't we tickle ourselves? So they developed this machine to do the experiment, and the experiment is an MRI study. They developed this system to do the experiment. And this is the subject's hand. And this little pin here is something that go and make touch like that in your palm. And you feel it. Right? And the subject itself can cause the movement. The experimenter hands can cause exactly the same movement. And then we can wrap this around, and then you can do the movement without any sensory input. So we want to compare the feeling, or not the feeling, the response of the brain under these 4 different conditions. When the experiment tickle you, when you tickle yourself, when you do the movement without tickling, in order to separate all these things. And what they found, that if you look on the somatosensory cortex, we're looking at the somatosensory cortex, externally produced tactile, that's where the experiment, somebody is tickling you, you have a huge response. When you tickle yourself, nothing. The somatosensory cortex is not responding. Then they looked to the cerebellum, and they found self-produced tactile signal produce huge signal. So I know that when I do it to myself, I know exactly what to expect. I know that I'm going to take, I know what the sensory things will be like, and this response is used to basically silence the sensory area in the cortex, saying, come on, this is not important. I do it to yourself. It's nothing about the outside world. The brain is there to tell you something about the outside world. This is not outside world. So the bottom line is that according to this and many other studies, the role of the cerebellum is prediction. You have to predict what's going to happen. And prediction, in my view, is the essence of brain function. We don't do anything other than prediction in our brain. We can do everything else without the brain, but we cannot do prediction. Prediction means you take the motor control, your movement, and you have to subtract it from the overall sensory information. You need a system that can do that. That's what the brain can do, and we are going for a break. What is that? Sarah? There, look at the
אני נשמע מוצלח. אני גם מתכנת חנרה. פעם נשמע בנקסטות לתקן. אני מעבר אותה?
הנה, בשבע.
בדרך כלל אנחנו יותר עבר אנשים גם ככה. עד שהם חזרו משום פקידה, אז אני כבר יכול לעזור אותם. אצלם בפה. לא?
אוקיי. יש לי משתנה אחד שהוא שווה לדלתא, שהוא שווה לאחד, אחד חלקי
אז אם אנחנו הולכים בעל פה.
עוד משתנה שאנחנו כופלים בו שדה. המשתנה הזה, כמו שאני מוסיטה, שווה ל-F, כאילו לאחד כפול של F ושאר זה כפול זה שווה למשתנה הזה, של אחד בחזקת... כן. כי אני מוסיף את שתי משתנים האלה, אחד בדלתא פאקשן, אחד בדלתא שניהם ביחד. כאילו, אחד או מינוס אחד או...
זה קשה, כאילו, טוב שלי לא פרחה...
אחד, כן.
סלאם זה... לא.
מינוס אחד זה שווה ליחידה, גם.
בעלי חיים? מנגינה? בתחת. על בעל חיים?
וגם אתה יכול לחשוב
משחה.
אני רואה מה יכול לחשוב דברים כאלה.
זה קרה.
אחד חלקי שתי פי, אז אחד חלקי פי, אחד חלקי פי. לא בטוח גם לגבי נקודת זה נכון. בדלתא U, A, I, J, שאני יכול לדעת. כאילו, for a given U, for a given U.
לא משנה, אבל שזה אומר מתי שזה שווה לזה, זה מקבל אחד.
כן, זה משהו. אבל השאלה איך זה עובד, כאילו מה איך זה כאילו, for a given U, אני
אומר U זה סיגמה.
כן, אבל גם
איך העץ המים לא משהו שהוא לא יסיגנל במילוי, גם עץ המים יכול להיות.
קודם כל את יודעת את יש טעות מסוימת שכל ה-AI
עשיתי כבר פעם אחת כזה...
אני חושבת שהסימן סימטרותי, אלא יותר העניין שיש את זה.
אני לא יודע אם זה נכון.
משהו משתנה שם.
למה הייתי בודק
זה מה זה מאוד, שהוא פועל. אבל אם יש קונפליקטר הבן, הוא יצא כזה טקסט. זהו, פסנו.
נכון, אין אף יד נכון
לא פוספטי?
פשוט יש שם משהו לא ממש מסתדר לי עם החפיפה שלו.
טוב, זה גייט.
לא, אני בדיוק משאר
למה?
תשאירו את הדברים באנגלית. תשאירו את הדברים באנגלית, לא יודע מה אני מדבר.
זה הוא מזלפתי כבר, אני לא יודע שאתה אומר.
רושמת באלטק, רושמת באלטק, Physiology A, 2017, 2018.
גו משהו. גוני?
קטע מתקשר למשהו מוכן להוציא. מה? תשעים אחוז.
Okay, we can, there's no problem on stopping in the middle to ask questions. It's more a discussion than a lecture. So we cannot tickle ourselves because the cerebellum provides a prediction, and the meaning of this prediction is to the brain, ignore, it's not important, you generate it by yourself. But it's not only motor. If you think about prediction, what you have to calculate precise timing, or to predict precise timing when the sensory input is expected, because you are generated. So you have to translate your movement into a signal that's saying, now you're going to feel a touch or a tickle or whatever. So you have to predict it. You have to generate well-defined timed signal. So time is the essence, as I said at the last minute, is the essence of all brain function. It's not something particular to the cerebellum. In the cerebellum is more pronounced because we know it's related to coordination and prediction, then time become an important. If you think about cognitive function, so it's you'll be delay in 200 milliseconds or 100 milliseconds, who care? But if it's a movement, and you have to close your eyes before a fly will fly in, then the 100 milliseconds is crucial. So timing is all over, but when it got related to motor task, it's become a matter of survival, timing. But it's not only timing. There's not only motor, so it's timing, it's not only motor. And I bought this paper, and this paper did a MRI study. In an MRI study, they asked the patient to do these two types of tasks. What they have to say is this TN interval is larger or smaller than the interval before. Sound. It sound tick, and they have to say if the interval here is larger or smaller than the interval here. And there are two types of signal. One is, you see, the interval is variable, and you want to compare the last and the one before it. And the other one, the interval is constant, and you only the last one is different. So you have two types of timing. One is that you have to look at it at the background of regular signal, and the other one is absolute time. You have to compare this interval to the one before. And then we found significant activation in the olivocerebellar network comprising the inferior olive, vermis, and deep nuclei, including the dentate nucleus, during absolute duration-based timing. This is absolute, only compared to this or that. And a distinct network for relative bit-based timing, which is that, incorporating a striatothalamo-cortical area. So you have two systems that use to estimate time. One is absolute time, because you don't care what happened long ago. And the other one is that I see it's not as a timing, but as something different. You see a difference. You don't see timing. Here you see absolute timing. Here you see a difference. And they conclude, and this is the MRI study, and you can see a sagittal section through the cerebellum and the brainstem, and this is the area of the inferior olive, and the deep nuclei are here, and a lot of cerebellum of the auditory part, the back, volume 10, 9, and 10 that are involved in vestibular auditory responses. They are turned on when you do this task. They don't turn on when you do this task. So they concluded the inferior olive and the cerebellum acts as a precision clock to mediate absolute duration-based timing. So the system is capable of generate or estimate absolute time interval. And when I said to generate absolute time interval, it means that you can be very accurate in this is the accuracy here is in the range of tenths of milliseconds or even less. So you should be able to generate signal at extremely high accuracy. As the inferior olive and the cerebellum acts as a precision clock. And that's what I want to talk in the next couple of minutes. What's the meaning of a clock? What do we need for a clock? In this clock we need a battery, but before the battery we have a spring. And before the spring we have a pendulum. We need a pendulum. That's what we need. So when you look into the olive, and what you find out, that the membrane potential is oscillating in a sine-like wave, very much like a pendulum. Every now and then we have an action potential generated. And these are the recordings from two cells simultaneously. And they both doing it exactly the same. This is basically a pendulum. You have a very clear interval in time. And then you can multiply it, and you can divide it, and you can do many things so that, but basically it's a pendulum. And a pendulum provides time signal. I just want to make a small detour, because if you look at the action potential of the inferior olive neuron, it's not a regular action potential. It's not a signal and that's it. And I just summarized the basic properties of the action potential in the inferior olive. So if you look at that, if it was a simple sodium-dependent spike like what we have in Hodgkin axis, it would go this way and then go down to the dotted line, making an after potential and go back to the resting. This is the classical action potential. Inferior olive action potential going down, but then it stops. There's a slowdown of the repolarizing phase. Then it becomes fast again, and then it stayed down there below the original level for a long time, and then it go up again and starts to fire again. And there are many, many different ionic channels that contributes to this signal. And one can think about, okay, just why do we need it? Why do we need here an action potential that is 20 milliseconds in duration, 10 to 20 milliseconds in duration? What do you need it for? And a more recent study shows that this is what you see in the cell body of the olive neuron. But once this thing is propagated down the axon, it generates a burst of action potential. So you can deliver the information on the width of the spike by the number of action potential you have in running up the axon. And this action potential will go all the way to the Purkinje cell, making the climbing fiber strong synapse even stronger. But this is not the point. The point is, if you can modulate the number of spike running along that very wide action potential, you can add information to do whatever is needed in the cerebral cortex. So from a biophysical point of view, we have this thing, which is the sodium-dependent action potential like Hodgkin-Huxley. That's what we would expect. We have calcium action potential that's mostly located in the dendrite, and it generates this very long thing. Now, calcium is rushing in once these channels are open. Calcium is rushing into the cell. The increase in intracellular calcium opens a specific channel, which is called calcium-dependent potassium channel. I hope you learn it. If it's not, you should. And this opening of potassium channel, which is calcium-dependent, will hyperpolarize the membrane and generate all this very long thing. The point of the olive, we have another kind, a low-threshold calcium signal that is working around the membrane potential and below. And this long hyperpolarization is sufficient to activate this other current, which is low-threshold calcium. And if this is big enough, it will generate another action potential. So you have a kind of a mechanism to generate rhythmic activity on a single cell level. But this rhythmic activity is associated with action potential running periodically in the fluorine. But most important, as we saw in the first slide, the membrane potential is oscillate like a sine wave. It's oscillate at a range of 5 millivolts in amplitude, and in this case about 3 or 2 to 6 hertz. In this case, it's 3 cycles per second. And all the record that I'll show you are done in isolated system, in vitro system, and also in a recording from the whole animal while it behaves. This can go up to 8 or 10 hertz, right within what's known as the theta rhythm, and the entire brain is working on theta rhythm. I don't know what theta rhythm is, but it says to say something about the frequency, but the mechanism of generating theta can be completely different in the brain or in the inferior. So the first thing which you want from a clock, you want the clock to be stable. Because if it's lousy clock, it's going to go fast and slow all the time. It's not good. So if you look at this oscillation, and we look at that over we make a measuring the autocorrelation at a long period, and you find that the frequency is stable over 2 minutes. Very stable. In this case, it's about 3 hertz. very stable. It can go on and on and on without, it is not disturbed, it's going and go on forever. So we have a stable clock. The question is, how synchronized is the clock? Because, you know, in a single cell in the entire brain making, who cares? Nobody cares if a single cell. There's got to be some sufficient number. If you look at that, on synchronicity, measured from two cells, as we saw in the Bisping, you find out the cross correlation. Beautiful. They tick together like a Swiss clock. They're doing it beautifully. So we have synchronicity. Next question is, what about the temporal resolution? You know, let's say even theta rhythm or 10 hertz. At 10 hertz means that the interval is 100 milliseconds between the peak. That means that this is the accuracy of the system. 100 milliseconds is not enough. We need better than that. How you generate better than 100 milliseconds if the cell oscillating like that? So occasionally, you find out that two cells, they oscillate off phase. So you have two cells that oscillated off phase. Now, if you take into account phase differences, you increase the accuracy of the temporal code, huge, are basically unlimited. So you measure from many cells, you can see all kinds of oscillation and that, and then you want to check the stability of the phase relationship. And you make this measurement of stability of the phase, and the red dots here represent this experiment. The yellow dots represent the experiment previously, which have exactly the same phase. Then you have a zero phase difference. And this blue thing is interesting, representing this experiment, where you have beating oscillation. And you look at this area at large, you see that you can have all kinds of phase difference during the cycle that cover basically the entire time range that is needed. So we have the rhythm by the oscillation, and we got the accuracy by the phase differences. Now, this is very nice, but we want to reset the clock, because we want to start measuring time from a given signal. Something is coming. You know, we want to start measuring it, like a stopwatch. You have to start the watch to start ticking. Or you have the watch is going wildly and you have to reset the thing. So we'll study resetting using this kind of mutant animal that enable us to use optogenetic to activate specific input to the area. And you got this, luckily we have this mutant is very widely used in neuroscience. And you look on the inferior olive, and this is the inferior olive. It doesn't look like olive. Does it look like olive? Maybe. The inferior olive is not labeled, but all the input to the inferior olive are labeled, so you can turn on the light and activate input into the olive. And sure enough, if you turn on the light, you make record from a single cell. You can see the response of the neuron to the light that we introduce. Sometimes the input is strong enough to generate an action potential in the inferior olive. And as you can see, the action potential is very much the same as I described in the previous slide. Very broad action potential generated by a single synaptic input. Now we turn on the light, when the cell is oscillating. And what you see is that you turn on the light again and again and again, and it doesn't matter what was the phase of the light, the phase relative to the oscillation, after the light that got synchronized. It means we reset the oscillation by the light pulse. Each time the light is coming, he starts the oscillation from a certain phase. So we have a system to restart the clock. And we want to quantify a little bit this resetting system. So these are the oscillation, demonstration for the oscillation. This is the light pulse. We measure the time of the light from the last wave and the time for the next wave. And we plot it as a function of the, not absolute time, but relative time relative to the period of the oscillation. And then, if we have a total resetting, we expect the red one, which means that T2 over 2 is independent of the oscillation. It always have the same value. It will start the oscillation. That's the meaning of a horizontal line. If we get the black line, it means that the stimulus doesn't do anything. So the oscillation is not unrelated to the stimulus onset. So with this quantitative ability, we want to reset the clock. So with this example, we look. There's one advantage of using light to activate a specific. I'm sure you heard about optogenetics. Nobody can finish high school without learning about optogenetics. So the advantage of the optogenetics is that we can control the area that we turn on the light, and we can activate this more or less input into the system that we are interested in. So if you take and turn on the light on the entire field of view, and these are the oscillation, and you turn on the light and you see perfect resetting of the clock by the light. And indeed, our quantitative measures show this beautiful horizontal line. Then we turn on the light only on this corner of the visual field. When you turn the light only on this visual corner, there's nothing. Then nothing happened, and the oscillation and our quantitative analysis show exactly what to expect when it's nothing. Now we do an interesting thing. We can turn on the light along this area. So we have a full area. We have only the right, lower right corner, and we have only the right side. And when you do that, at the very beginning, when it's on the falling phase of the waves, it seems to be working very nice as generating precise resetting. But if you go down to this area, it suddenly jumps to a different thing, resetting in a different way. And finally, if you go to the last part, it has no real effect on the phase of the response. So what's the meaning of that? Meaning that it depends what input you activate or deliver to the olive. You can completely reset the clock. You can partially reset the clock. You can reset the clock only for the very first part of the wave. It's very accurately measured. The ability to change the clock, to reset the clock to different intervals, it means that we can really control the oscillation, and by that the timing they will deliver by the inferiority. So we can reset the clock. and we can do it very nice. We can do it precisely in millisecond range to reset the clock. So, now what's left? We have to start and stop the oscillation. Because, you know, we go to sleep, you don't need any oscillation. We do have very slow, but we don't move. We better not move. We better not pay too much attention to our dreams. It's not very healthy. It's nice, but it's not very healthy. So we don't need it. We have to stop it. We need it very strongly on active phase of our daily life. So we need to start and stop. So how do you start and stop the clock? You cannot hold the pendulum and say stay still. So the neurons in the inferior olive, they are organized in a network of coupled neurons. So two neurons, got together. There is an electrical synapse between the two neurons. An electrical synapse, I'm sure you heard, is a kind of a synapse where current can directly flow from one neuron to another. No need for chemical transmission, no need for release and probability of release. It's direct current going from one cell to another. There's a big war of electrical synapse over the years. Basically, all scientists, you know, they like to fight. I don't know why, but they like to fight. So, but if you think about it, 100 years ago, Golgi and Cajal, they fought are the cells are separated or not. And it was a real fight. And then Cajal won because there is a gap between the neurons. But then came, after that, Otto Levy and introduced the chemical transmission. And he did it. It's a very strange experiment on a frog, and he delivered the blood from a frog to one to another. And basically, from experimental point of view, this experiment cannot succeed in any way. Anyhow, it took him many years to fight to introduce chemical transmission into the study of the nervous system. It was a huge fight. And then, in the last 30 years, suddenly they rediscovered the electrical synapse. Again, the fight starts again. Because it took as many years to introduce chemical transmission, now it takes as long to introduce electrical transmission. And if somebody is interested in the history of neuroscience, I would recommend a book called The War of the Soup and the Sparks. And the soup are the chemical transmission, and the sparks are the electrical transmission. He gave a beautiful account for all the history from the time of Cajal and Golgi up to this day of the war of the soup and the spark. Anyhow, in the inferior olive is one of the first brain area that show the electrical synapse. Today we know that most of the cortical interneurons, all the GABAergic neurons in the brain, are connected by electrical synapses. And here is the first thing to show that there is electrical synapse. He somehow related to the oscillation. I will get to that in detail later. So the electrical synapse supports rhythmic activity, and the electrical synapse supports synchronization, that's for sure, because it's direct current from one cell to another. There's no synaptic delay, there's no jitter in the synaptic delay. It's perfect synchrony. That's what is provided by the electrical synapse. Perfect synchrony. So now to this day, we know that all the PV neurons are electrical coupled. It is interesting that the electrical coupling is only between neurons in the cortex, is only between neurons of the same type. If you know something about the cortex probably here, it's you have power valve neurons, only the PV neuron connected to each other. Yet you have somatostatin neuron, also inhibitor neuron, only they connect among themselves. And recent work shows that electrical synapse can be modulated. Can be modulated exactly the same way as chemical synapse. You can make them stronger, or you can make them weaker. You can make them short-term change, or you can make long-term change. All this terminology exists for electrical synapse. And today, some of the research try to correlate this electrical network in the cortex to all the different rhythm that we find in the cortex, the theta, gamma, beta, delta, God knows what else. Everything is there in rhythm. Some of them is artificial rhythm, or some of them is real rhythm. And today's work saying, okay, attention is delta, beta, cognition is gamma, whatever. Sleep is delta, that I know. In the Physiology was one of the first mammalian systems that demonstrate gap junction between the neurons. So that neurons in the olive are coupled. No wonder that they oscillate together. But still we have some with different phases. We have to explain that. But the fact that they oscillate together is not surprising, because current can go from one cell to another. No question that they can be synchronized. So many years ago, they demonstrate that this structure, which in a more simplest way is described here, a neuron, one neuron is connected to another neuron through gap junction, and current is going from one neuron to another. That's the meaning of electrical synapse. Now, if we have a chemical synapse here, then instead of going out to the other neuron, it will go out through the holes made by the synapse. So in electrical terminology, we're making a short circuit. We prevent the current from going from one cell to another. Instead, it's leaking out of the cell. That's the meaning of a short circuit. So by that, we can actually functionally block the gap junction. Because if the current before went to the other cell and now it's leaking out, the second cell doesn't know that something happened in the first cell. So functionally, it's uncoupling the cell. The morphologically, we can see that a gap junction is what we see here, where the two membranes. This is one dendrite, is another dendrite of another cell, and this area here, the membrane are getting very close to another, and this is an enlargement of this area. You can see that basically the membrane of the two cells are almost fused, and they are fused by the channel that enable the current to flow from one to another. And in a more recent work, somebody went into a lot of trouble to demonstrate that this gap junction, as you can see, is surrounded by terminal with synaptic vesicle, and you have a chemical synapse here. So you have electrical synapse here, and a chemical synapse here and there. And somebody went to a lot of problems to demonstrate that these synapses are most likely inhibitory synapse, and most likely originate at the deep nuclei. If you still remember the deep nuclei of the cerebellum. So what we have to do is to label the neuron with optogenetic stuff, and in the deep nuclei, and those GABAergic neurons that go to the inferior olive, and we look what happened in the inferior olive, and indeed, when you look at this area here, you can see the cells are beautiful, labeled. Only the GABAergic neuron, you know, the big thing about the optogenetic is extremely specific. So if you want to label neuron, you can label only GABAergic neuron, only neuron that express PV, only the thaumaturgy. You're very, very specific. This is the genetic part of the opto. The opto is that you can activate or inhibit the cell by light. So you can see that the cells are labeled, and if you go down to the inferior olive, you see that a lot of axonal terminal there expressing the channel rhodopsin, and the only thing we have to do is turn on the light, and we specifically activate the connection between the deep nuclei and the inferior olive. And of course, if you do that, you turn on the light, you can see you have a membrane potential go down as expected from inhibitory synapse, and have a reversal potential of chloride as a typical GABAergic synapse activated by light. That's what we wanted to do, to activate this synapse. And we succeed in activating this synapse, a pure inhibitory synapse, climbing from the cerebellar nuclei into the inferior olive. Now what happened? So we measure from two neurons. This is basically the way to characterize the efficiency of electrical synapse. You inject current to neuron 1, you measure the response of neuron to this negative current injection, and then you measure the response of the postsynaptic neuron to the same current. So inject to neuron 1, you got the response in neuron 2. And you inject the current to neuron 2, you got the response in neuron 1. Current is directly going from one neuron to another, and we define and then we activate the GABAergic input, and the response on the postsynaptic neuron becomes smaller in both directions, meaning that we were able to reduce the coupling, the efficiency of coupling from cell 1 to cell 2. Because some of the current is leaking out through the open GABAergic channel, we managed to reduce the coupling. And we can show that in different neurons it's happened a lot, and it's extremely effective. Now you take this neuron, and you turn on the light, and the oscillation stops. So you have oscillating neuron, you give a pulses of light, and the oscillation stops. And you can go longer with the pulses of light, or even longer, and as long as you have the activation of this input, we stop the oscillation. So we can stop and start the oscillation by the activation via the cerebellar nuclei. To remind you, these cerebellar nuclei are innervated by Purkinje, so I can control the on and off of the oscillation by the input from the deep nuclei.
The neurons are so strongly coupled with one another by gap junctions. How do we get the fading? You showed before that we had these all מקרים שהם היו ממש מוכנים.
Maybe today, maybe next lecture. Very good. Anyhow, this thing is as before, it's a very special, different. So we can have a recording from all the neuron, and we turn on the light, and we can see we stop the oscillation. And we did what we did before. We turn on the light only here, up there a little bit. Then we turn on the light here, also nothing. Light here, complete blockade of the oscillation, and light here, also complete blockade of oscillation. So by activating a specific input, we can control how much of the olive is going to oscillate and how much is going to be quiet, and we can do it in a very precise way. So we want a clock to move our finger, and we want a clock to move our thought, and we want a clock to predict sensory income. We need several clocks. So we can have these clocks within a group of inferior olive neon, and we'll get to that question in a minute. Okay, this is the minute. The question is basically what makes the oscillation? How do you generate oscillation? What do you need to generate oscillation within the inferior olive neon? We know that is a properties of a network, and as well as the properties neon. Each one by itself is not really can do it. We need both the network and the property. So many years ago, we composed a very simple model. We want to see what's in the network and in the neon that generate the oscillation. So we composed a very simple model. In our model cell, we have only calcium channels and leak channels. Calcium conductance and leak conductance. And the calcium conductance is a very special calcium conductance found in the inferior olive and in some thalamic neurons and many other places today, where the channels are inactive at rest. Then you have to hyperpolarize the membrane. By that you remove the inactivation from the channel, and then when the membrane goes back to the resting, it can trigger the response. It's called the low threshold calcium. Most of the high threshold calcium does not have inactivation function. The low threshold have inactivation function, and therefore, first, it's very sensitive to the membrane potential, and second, it stops by itself. So we take this model, and you can see that the calcium conductance follow Hodgkin axis like dynamic, and this is a constant, and we want to see what this kind of cells, how they will respond given that there is a heterogeneity in the density of channel. That's the only difference between the cells. They have more or less leak or calcium conductance. So you have a network of heterogeneity cells that they don't have different channels, they just have different density of channels. So if you look at that, and you look at the calcium conductance on this axis and the leak conductance on this axis, you can see that you can divide the neuron into several groups. All the neurons are, with these parameters are defined as stable. I mean, they respond to current injection in a stable way. You inject current, they have some regenerative response, and you inject negative current, and so on. Stable cell. All these cells in this area where you have a high calcium conductance and a very low leak conductance, we have conditional bistable, which means that the cell either in this level or this level, it will go back to bistability in next talk. In between, yes, conditional oscillator, which means that you have to hyperpolarize the membrane, and the cells start to oscillate, and only a very small area are cells that are spontaneously oscillate. So we have all this kind of behavior depend on the different density of either calcium or leak conductance. That will give us this thing, and the result is, I skipped this one. The result is that if you take a group of neurons within this area and you couple them with electrical coupling, they will start to oscillate. And it doesn't matter, each one of them by themselves are not oscillating. When you couple them, they will start to oscillate, provided they will fulfill two basic roles that we will talk about them next talk. Thank you.
נפגוש אותך ב-11 במשרד, טוב? או אתה רוצה קצת אחר? 11 ורבע?
אני גם לוקח ממנו תמיד שאני צריך. זה כמו בנק. זה כמו בנק.
זה מזכיר שגם אני חייבת לך.
איך היה? למה לא באת בשבת?
אמרתי לך.
למה? אני ישן. זה לא סיפרת לנו.
אין לי עוד. אין לי עוד סיבות. הייתי ממש קרובה, אבל סיימנו ב-4, ואז מה? אני אחזור, נלך, מה למדתם?
שיעור על מרקר. מרקר?
מה?
הרגעת? מרקר?
מעקה.
אוי, זה לא את כאילו את נותנת את השאלות? לא, שיעור כזה. ואנחנו צריכים עד פעם הבאה. זה שיעור. כן. זה מאוד יפה, וזה נותן סטיות ברמה וזה. זה כאילו מעלה לך את הנושא של המצווה. וכאילו מבין שיש לו מיני סטיות שכאילו מכוונים אותך לחשוב על המצווה בדרך יותר עמוקה ממה שחשבת. זאת אומרת,